So we are going to be doing something a little different today. We're actually going to be taking notes together from one of the lessons. If you've already gone through this lesson, I'm going to be adding extra information that you may not have known. And if you haven't gone through this lesson, it's going to be super helpful for you because you're going to be able to take notes, and I'm going to save you some time for doing that. I really just want to show you guys how I would pick out important information, because I know some of these bio lessons have pages and pages of stuff, and sometimes it's very confusing to figure out what to actually write down versus what you can ignore. So I'm going to try to model that a little bit. And I got a document camera. I'm super ready, and I have a humongous coffee, so we're good to go. So I did make a PowerPoint. Let's see. Okay. So we're going to be talking about the Module 2, Classification and Diversity. Most people are finished up with Module 1 or finishing up, right, the evolution module. So we're going to be moving on to this one. Now, in my personal opinion, this is a pretty easy module. There's nothing groundbreaking or crazy here. It's all about how we organize living things and what traits they have and learning about the different kingdoms, basically. It's pretty interesting. You're definitely going to learn some stuff you probably didn't know about the different kingdoms. But it's nice to have a little break after the first semester of incredibly hard material. So keep that in mind. All right, so what we're going to do is we're going to describe the systems scientists use to classify organisms. We're going to differentiate between cladograms. We're going to talk about those because they can be a little hard, but they are super useful in terms of looking at evolution and classification at the same time. And then we're going to practice picking out key information while note-taking. So, classification of living things. If you did this lesson already, I want you to get your notes out, right? Whether that's a document, a piece of paper, whatever you did. If you didn't take notes, well, then you're going to take notes now. And if you haven't gone through this lesson yet, great. Get out some paper, get out a document if you want to take notes virtually on the computer. I'm going to be taking notes on paper so that you guys can see what I'm writing and how I'm organizing. So, all right, I was like already in this lesson because I kind of already started. So I could do some talking during this. Okay, so the nice thing about this lesson is that the first page is pretty useless. It's some boring game about organizing stuff. In fact, I think they use this in physical science as well. I didn't even do that. I just went down here and said whatever and read the objectives. So after completing this lesson, right, this is an important step, I think, in doing any lesson online, is actually looking at the objectives. That's what you're supposed to get out of this. They don't say anything about serial or organizing it, so I already know I don't need to know that stuff at the top there. But we're going to be describing classification as a work in progress, discuss the characteristics of the 3 domains, bacteria, archaea, and eukarya. Ugh, that's such a hard one to say. Describe classification by cladistics, summarize how molecular evidence reveals species relatedness, identify the structures and shapes of viruses, and describe different types of viral infections. So we're going to talk about viruses very briefly, but again, this is a weird thing they did here. Viruses don't really belong in this lesson. Viruses are not living, right? And even though they contain DNA or RNA, they're not considered living things, which we'll talk about when we get to viruses, but they do interact with living things pretty heavily. All right, so we're going to the second page here. I am going to figure out how to do this the best. Let's see. If I make this a little smaller, let's see, this, put at the bottom maybe. I wonder if I can split the screen. Split screen. Oh, no, that's not what I wanted. Whoa, that's cool, though. Okay. Sorry, I literally just learned how to use this document camera last night because it came with a CD, which I can't even put CDs in my computer. So I was like, great, how am I going to do that? So I had to download the software. It was really annoying. All right. So first things first, I titled my page here. Why isn't this loading? This might be harder than I thought it was going to be. We might have to go back and forth. So I titled my page here, Classification of Living Things, right? They say living organisms, whatever, same thing. And I put the date, right? Just because it's helpful to be able to come back and figure out when you were learning something sometimes. You might remember like, oh, it was winter that we were learning that. That could be helpful. All right, so first things first, I'm going to read to you guys. Nice early reading in the morning. So during his study of plants, a Greek philosopher and scientist named Aristotle recognized the need for a classification system for living things. That's about 2,300 years before the discovery of DNA and approximately 2,100 years before Darwin postulated his theories on evolutionary changes in organisms over time. Since Aristotle did not have the scientific knowledge we have today, he created a hierarchy of life based on what he knew about organism complexity. It was called the Great Chain of Being. It placed humans at the top since he considered them the most complex organisms in the world. Beneath humans, he placed 10 less advanced species. Interestingly enough, he places plants at the very bottom, although they are highly complex organisms in modern terms. As scientists discovered new organisms, there was an increased need for a new way to organize them. In 1735, Carlos Linnaeus, a Swedish botanist, which is also a guy that studies plants, created a new classification system that sorted organisms by their physical traits, not their complexity. A modern version of this classification method, Linnean taxonomy, is used today. All right, so we're getting a little background here, a little history of how this system came about. So is this big enough for you guys to see? Yes? Okay. So for the history, I just made a little note here. Aristotle, I probably should have capitalized his name, whoopsies. Aristotle pretty much was involved in everything. If you ever study history of any science, Aristotle was there doing something. Even math, he was there doing it. So not surprising, he was like, oh, we should organize stuff. And also not surprising that he said, oh, well, humans are clearly the most complex organisms because we can think and we can classify things. And then he said other organisms are below us, and then plants, like what are plants? They're at the bottom, right? And it's interesting because this was a pretty common train of thought, I would say, all the way up until the 1960s and 70s. Pretty much most scientists would agree that humans were the most complex, right? And then they did this study where they actually analyzed the amount of DNA and the complexity. And most scientists hypothesized that if you're a more complex organism, you're going to have more DNA, right? That would make sense. The more DNA you have, the more complex, right? The more things that are going to code for whatever you need to be complex. Well, we found out that that's not true at all, right? In fact, ferns, the most basic plant of all time, have 1,350 something chromosomes. Right? We have 46. Meanwhile, Kendrick, if you look at a fern, it doesn't seem complex, right, compared to a human. So that kind of destroyed Aristotle's concept here. So in fact, I'm going to write that on my notes as well, that amount of DNA does not equal complexity. Right, and that plants usually have more chromosomes than we do, which is pretty strange to me. When I first learned that, I was like, whoa. So that's what I wrote here for history, right? That Aristotle made this great chain of being. Somebody at least was thinking about organizing living things. You guys are going to be answering an exit question at the end of this, like, why would we even need to organize living things? This may seem sort of like a unnecessary step. And it's possible you could argue that we don't need to. So then we start looking at this dude, Linnaeus, called Linnean, I don't know how exactly to pronounce it, I think Linnean, taxonomy, right? This is the way that we have been doing it for many, many years here. In fact, you might have heard of this before: kingdom, phylum, class, order, family, genus, species. Right? When we come back over here to the lesson, and we scroll down a little bit, we see here that Linnaeus divided nature into three kingdoms: animals, vegetables, and minerals, right? So he kind of started off a little bit more vague, right? Mostly because they didn't really know how many categories there might be, right? And he kind of wanted non-living things to be in there, like rocks and soil he thought should be part of the classification, which obviously now they're in their own kind of area. And then really when we started studying things and realizing that animals and plants can be divided into smaller things, right? Like starfish are technically animals, yet they really have not that much in common with humans. They have more in common than you think. But when you look at one, you're like, oh, that's in the same category as me? That's weird. So we learned from Darwin that animals do change over time, right? And therefore, the Linnean taxonomy system has also changed over time. Makes sense. So basically now we use these 7 major taxa, is what they're called. Make sure you have those written down. Kingdom, phylum, class, order, family, genus, species. Kingdom being the biggest, species being the smallest, which is why they showed it in this circle thing. I just listed it out. So this is a helpful thing down here. Did you know? So a mnemonic device, in order to remember this, right? You guys have the luxury of being able to use your notes pretty much at any time. So you may not need to memorize this, right? But when I took biology, I had to memorize this, and I had to memorize all of these things about all the different phyla. It was like ridiculous. And I don't remember any of it now, really. I remember random pieces, right? But it is helpful to kind of just have this in your mind. So if you want to memorize it, this is one that they came up with. You could come up with your own. King Philip came over for galloping stallions. All right. So then they do this thing where they have these tabs. So the kingdoms of life. So they kind of get into this. I say they because I didn't write these lessons. So it's fun for me to critique them. But they kind of try to do this organization here to show you the different kingdoms and kind of what different features they have. But this to me is a little confusing in terms of how it's organized. So I'm going to show you guys my way of organizing this. All right. So we have our 5 kingdoms. And what I'm going to ask you guys to do is I'm going to write down notes as I go through this. I want you to write down notes as well. This should be pretty self-explanatory because I already kind of gave you the important information here. But regardless, we have our first kingdom here, kingdom Archaebacteria. These are the extreme, crazy bacteria that live in volcanoes, live in places without oxygen, live deep in the earth, right? Possibly live in space. They are pretty intense and extreme and therefore in their own kingdom. They are unicellular, right? Which means they have one cell and they're prokaryotic. Does anyone remember what prokaryotic means? This was from like module 1. You can type it in the chat, or if you want to speak, you can. What is prokaryotic? The DNA is in the cell, not the nucleus. Yes, that's definitely part of it. Anything else you remember about prokaryotes? They're simpler cells, right? They don't have membrane-bound organelles, which is why they don't, excellent, lacks a membrane-bound nucleus. So they don't have a membrane around the nucleus, which is why the DNA just floats around. They don't have any of the organelles that have a membrane. So they don't have mitochondria. They don't have chloroplasts. They don't have lysosomes. They don't have like a lot of these fancier organelles. They basically just have ribosomes and DNA, and that's it. Kendrick, stop. So these are the simpler types of cells, right? These were most likely the first cells. And the word literally means, from Greek prokaryotic, it means no nucleus, right? All right. So eubacteria is the other kind of bacteria, right? A good way to remember it is that eu sounds like you, meaning the bacteria that can affect you. You'll probably never encounter an archaebacteria in your life. Although if you become a scientist and you go exploring caves or something, maybe you will. But we have our eubacteria. These are the more common bacteria, but they are also unicellular and they're also prokaryotic. This was actually a big discovery back in the day. I don't even remember when it happened, but scientists used to just think all bacteria was the same, right? And no one really studied it that much because they were just kind of like, bacteria is bacteria. And then they discovered that there's serious differences between eubacteria and archaebacteria. So much, in fact, that they're in different kingdoms, which means that they have more, like, as many differences as we have with plants, right? So it's pretty significant. Then we have Protista, which is sometimes referred to as the catch-all kingdom, mostly because this group is for anything that we didn't really know where to put it. So scientists were like, cool, let's put it in this category called protist. They're weird. They can be unicellular, but they can also make colonies. So they can look multicellular even though they're not multicellular, meaning that they're still single cells, but they're huddled together making a colony. For example, slime mold, this disgusting yellow stuff you're seeing here, right? That's an awesome example of a multicellular looking protist. It's really single cells all living next to each other, kind of like bees or ants or something like that. They are eukaryotic cells though, right? Which means that they have membrane-bound organelles. Their cells are exactly like our cells. They have mitochondria. Some of them have chloroplasts, right? They have a nucleus. And examples are slime mold. A lot of the different kinds of really disgusting molds are protists, and also algae, right? Big misconception that algae is a plant. It is not, right? It's a protist. But it acts like a plant. It photosynthesizes, it has chloroplasts, it has chlorophyll, right? It looks like a plant, but it's not a plant. Pretty weird stuff. Does it benefit them by making colonies? Yes, right? Which is exactly why some of them do it. I haven't studied protists a lot, so I don't know exactly what benefits they get out of it, but I think a lot of it just has to do with they kind of can exchange nutrients between each other. So it would be kind of like if you had to live by yourself forever versus you have 10 people that you live near who can like give you food if you run out of food, or they can help you do stuff if something goes wrong. Then we have kingdom fungi, right? Which I wrote on here, not plants, which might seem silly, but they really aren't plants. Even though a lot of times people think they are, same with algae, right? Fungi cannot photosynthesize. They cannot make their own food. They're heterotrophs. They have to eat other food, right? They're also eukaryotic, meaning they have membrane-bound organelles, and they are multicellular, right? So that means that they, like one mushroom is actually an organism on its own. They do make their own crazy networks underneath the earth where multiple multiple fungi are communicating with each other. They're pretty scary, honestly. Fungi creep me out. I think they're going to take over the world one day. They're like alien-like things. Very smart. They can live on dead stuff. They're really creepy to me. So I put lots of pictures of them. This red mushroom is like the most poisonous mushroom ever. If you ate that, you would die, right? They can make little stacks of mushrooms. These are those gross kind of mushrooms that grow on your lawn when fertilizer's there. And then fungi can also look like mold, where it's growing not in a mushroom shape, but more in a flat mold-like shape. So they're pretty, as I said, they creep me out. I know that's kind of weird, but they do. But they're pretty cool. And they're super important for the ecosystem, right? They are huge decomposers. And without them and bacteria, we wouldn't be able to survive. Then we have kingdom Plantae, or plants, right? It's the second largest kingdom, animals being the largest. Plants are multicellular, they're eukaryotic, meaning they have those membrane-bound organelles, and they're autotrophs, right? They make their own food. You can think of it like the word auto means self, right? So an autotroph can make its own food. Troph means obtaining nutrition. Lots of fancy, weird words going on. So we do have, like this is just some general fern-looking plants. And then we have a Venus flytrap, right, which is still a plant because it is still an autotroph. It can make its own food, but it can also consume nutrients from eating small insects and worms, etc. And mostly Venus flytraps do that because a lot of times they can grow in poor soil where they're not getting nutrients they need. They can absorb those nutrients from the animals that they digest. So plants are a huge kingdom. You will do a lesson about plants and learn all different types of stuff about how they reproduce, their cell growth, what they need, etc. We're actually going to do, our next lab is going to be about plants. We're going to grow plants, and then we're going to change a variable to learn about plants. So I'm going to give you guys a bunch of different options, but we're going to grow bean sprouts because they grow pretty easily and they're pretty hard to kill, although I'm pretty good at killing plants, so I could probably find out how. But they grow fast in like 2 or 3 days, so we can plant them and immediately see them. And then you can do different things like water them with Gatorade versus water. We're going to have some people do acid rain, so spraying them with acid rain that we make. We might try different types of soil, and we might even try light versus dark. And it's a pretty simple kind of experiment, but it's pretty interesting to see how hardy plants really are. All right, and then we have Animalia, which I didn't even put a picture of because I think we're all pretty familiar with animals. But there's over 1 million species, right? And they still discover species. If you Google new species, you'll see 2017, all the new species that have already been discovered, which is crazy that they're still discovering species. But animals are multicellular, right? And they're eukaryotic, just like plants and fungi. Sorry, I was trying to write the word eukaryotic. And they're heterotrophs, right? So they cannot make their own food, which we know because we have to obtain our food. So, just trying to get my notes up to speed here. Okay, and you guys can tell me if you hate this note thing, because then we don't ever have to do it again, but I thought it might be helpful. Okay, so that was my little spiel about the kingdoms that I thought was better than what they did here. But basically, they're doing the same kind of thing here in the lesson, trying to show you that prokaryotes, right, are single-celled organisms whose DNA is not within a nucleus. Eukaryotes have a nucleus, right, and they have membrane-bound organelles. So our prokaryotic groups here are archaebacteria and eubacteria, and then our eukaryotic groups are protista, plantae, fungi, and animalia. All right, last thing is this thing called binomial nomenclature. And I'm going to show you guys. So all I did for this is for each of the kingdoms, I just made bullets and I just wrote unicellular, prokaryotic. I didn't write examples just because I already know the examples, but you easily could have had examples. I also really like to highlight like headings so I can see them easily. Okay. But we're going to be going back here to this kingdom, phylum, class, order, family, genus, species stuff for a second, just because that's what binomial nomenclature is. It's a 2-word name that is Latin-based, and it is the genus and the species. So we might even want to write binomial nomenclature equals genus plus species, right? And it's Latin based. There are really specific rules. For example, the genus is always capitalized, the species is always lowercase, and it's always in italics. Weird stuff, but that's how it is. It's basically so that when you're reading through a scientific paper or an article or something else, you'll be able to quickly be like, oh, Felis chaus, that's some kind of organism, right? And if you're super knowledgeable, you'd be like, oh, that's a cat of some kind. So I would write an example of that in your notes. An example is Felis. How do you spell that? All right, Felis chaus, right? And that should be in italics. I don't know how to write in italics, so I'm not going to do that. And so, of course, the reason that we do that, right, is because here they say a single species can have numerous common names based on its location, the language, right? So especially like if you're looking at, like they're talking about salamanders, some people might just call those lizards, right? So if we want to be specific, we want to know the genus and the species because that's going to narrow it down to exactly which organism we are talking about. So here they give us some examples starting with order, right? So they eliminated kingdom, phylum, and class. And they show us that in the order Carnivora, we have 2 families, we have 3 genuses, and then we have all these different species, right? They give us some very different things. We have cats, then we also have a polar bear and a bear, right? Which kind of makes sense. If you think about cats and bears, you're like, oh, they're probably related, right? Doesn't seem crazy. So that's all binomial nomenclature is that genus and species put together, which helps us identify exactly which organism you're talking about and has nothing to do with language or location. Although it is in Latin, but still, no one really speaks Latin, so. And then finally, in the 1970s, we start having people say, oh, we should probably think about DNA and how it relates to organizing organisms, right? So this guy, Carl Woese, I don't know if he's German, Husa maybe, began to use nucleic acid sequences and ribosomal RNA to determine links between organisms. These links revealed that organisms with similar physical characteristics were not necessarily related to one another. In the case of the bacteria kingdom, ribosomal RNA uncovered the existence of 2 separate groups. Although they looked similar, they were different, right? So he kind of proposed that there's these 3 domains of life. So in my notes, I would write, you probably don't even really need that dude's name, but you could put it if you want. I would put like a little marker to demonstrate that we're in a different kind of section here. That we have these 3 domains of life based on RNA and DNA. So when they say ribosomal RNA, they're actually talking about taking RNA that makes up the ribosome and just analyzing the sequence, right? And we learned last semester RNA has pretty much the same kind of coding as DNA, right? Except that it has a uracil. So instead of seeing A, T, C, and G, we would see A, U, C, and G in that RNA sequence, and we would be able to actually figure out how they're related, right? And they can actually do that with you as well, because you inherited your mitochondrial DNA, because your mitochondria has DNA, which isn't RNA, I know, but it's a similar concept. You only get that from your mother, right? Because your mitochondria comes from the egg cell. So you can actually trace your ancestry using that mitochondrial RNA, because then it's going to trace the mothers, which is pretty interesting. All right, so we have these 3 domains, right? Definitely should write what they are. we have bacteria, archaea, and then eukaryota. Okay. So you could write the details of those, but notice, all I did was read all that stuff. All I wrote was 3 domains of life based on RNA and DNA, and I drew what they are. Right? It might be helpful to write a little description, but we already defined the kingdom, bacteria, eubacteria, and archaeobacteria, and eukaryota is basically plants, fungi, and animals, right, and protists. So really, we kind of already defined all that stuff. There's no need to write it again. All right, so did you know? We already read that. Okay, so that was fun. Now we're going on to page 3. How many of you guys wish that I just read all the lessons to you and gave you notes every day? So I'm needing that motivation sometimes, because I could feel you on that. Okay, so now we're going to switch into a completely different concept here of cladistics. So these cladograms. Now, for those of you who've already done the assignment, a lot of people were getting a little confused on these because they're kind of hard. But basically the concept behind this is that it's going to show us the evolutionary history of a group of genetically related species. So that sounds important to me that I should probably write that down. Phylogeny, if I can spell that. Oops, I missed a Y. So evolutionary history, of species, we'll just say, right? Because if they're having an evolutionary history, they're obviously genetically related. So this evolution of species is represented by a phylogenetic tree of ancestors and descendants, linking one species to another in a chain starting from the bottom of the tree to the top, right? So because we started using these phylogenetic trees, somebody was like, oh, let's do something called cladistics, which is a hypothesis. So we should definitely write that down as well. Cladistics is a hypothesis about how organisms are related. And the interesting thing is, I would even add in here, it's a visual hypothesis, right? Because you're literally showing how you think these organisms are related, who you think the common ancestor is, right? What traits cause them to branch off. So these can be really hard to make, right? Because it's a hypothesis. So technically there's like 4,000 ways that you could do it. And if you have an argument for it, then there's no reason that it can't be true, you know, until you start looking at evidence. But I'm even going to draw a few of these just so we have some examples. So this one's pretty complex, but it's okay. I'm going to draw just a few of these lines here so we get an idea. And then what they do on this is they actually explain why there is a new line happening, essentially, right? So we start with amphibians. They're just at the bottom because they don't have any of these traits that were picked here. The first trait that they picked is an amniotic egg. So that means that Amphibians don't have the amniotic egg. Everybody else has it, right? Because it happened here on the tree. So that means turtles have an amniotic egg. Then we add a new trait here to help sort of break this down further. So openings in skull for mussels. Okay. Openings in skull for mussels. And that's what's hard about this, is if you just get all these animals listed out and you need to organize them, you have to come up with these traits that you're going to use to separate them. And that's where it can get really hard. So openings in skull for mussels, turtles don't have that, but crocodiles, birds, dinosaurs, lizards, etc. do. Right? And then we have this group here, which is like birds, dinosaurs, lizards, and snakes. The next thing that we have that differentiates those is hair, right? Because none of those have hair. So you could draw the whole thing, but I'm just going to draw this one, where I just gave these two different things and show these organisms, right? And that this is a phylogenetic tree / cladogram that's really showing us how these organisms are related. And again, you have to pick those traits, right? When you're making a hypothesis, you have to figure out which traits are going to separate these organisms into groups. And that can be hard because you're thinking about it backwards almost. Instead of being told what groups they're in, you're thinking of the groups. So then down here, they give us this little description here. The more organisms in a cladogram, the more ways one could organize it. Besides making sure that there is one less clade, right, or branch of the tree, then there are a number of organism groups. There are no definitive rules on placement. So if the evolutionary links between species are in accurate depiction of ancestral and derived traits, the cladogram is correct, right? So usually you try to go for the simplest organization, but you might have something like this where it's just a line, and every single time you're adding a trait that the new group has. Or it could be more like this tree, which is really, if you're looking at a bunch of organisms that are really closely related, like for example, you're looking at insects or something like that, you're probably going to get something more like cladogram 2 here, because A and B are super closely related, but they're not necessarily related to C and D, E, F, G, H in the same way. If that makes sense, right? So these can be really hard to create. So when you're creating one for the assignment for this lesson, it might take you a long time to figure out how to organize them, right? And that's the point here, is that you have to think of those traits. Okay. So I can't remember if it goes into the viruses or not yet. We'll find out. Oh no, we have our alternate classification systems, right? So really, genetic connections, honestly, they just fall into all of these things, right? If you use genetics to make your phylogenetic classification or your cladogram, right, then it's 1,000% true because you're using genetics as your evidence for it. The Linnaean classification, right, is also genetic based. The genus and the species, those are really small groups that have a lot of DNA in common, right? So unfortunately, they say, though, this could be important, there are some disadvantages to using genetics for categorization. For some domains, like bacteria and archaebacteria, DNA is transferable through transformation, transduction, and conjunction. In all three processes, bacterial cells of like or unlike species exchange genetic material. So they're talking about here that bacteria can have sex with each other, right? That's what they do. They basically have an arm that comes out and stabs another bacteria and gives it DNA. It's kind of weird, but they don't do it all the time because they don't need to, but they can exchange DNA between species, right? Which is rare, because the definition of a species is that you can reproduce and create viable offspring, which means that you create an offspring that can also have offspring, right? So for example, a mule, right, is a horse and a donkey. A mule is not a species. Mules cannot reproduce. They're just there. Bacteria can exchange DNA with other species, which isn't supposed to happen. Like I said, they can break the rules a lot because they're pretty intense and they've survived millions of years, so they figured out how to do it. Oh, I should just share my DNA with this other bacteria and then everybody's happy. So because of that, genetics can be hard in terms of bacteria. It can be really useful for animals, plants, protists, and fungi though, because those organisms are not transferring their DNA as often, if at all. So I would probably summarize that in our notes here. I would say genetics is not 100% accurate because bacteria can transfer DNA between species. And then I'm going to write bacteria sex in parentheses because that's what it is. And if you don't know the definition for a species, you might want to write that down too, honestly, because it is a good one to know. And the word viable means an organism that can reproduce itself and is alive, right? First step being alive, second step being reproducing. So, all right, so we just kind of got a little bit of information here about genetics and maybe why or why not we should use it. To me, it sounds like probably not the best idea for bacteria, but for other organisms, sounds fine. All right, then we're gonna go into viruses. So this should be its own separate lesson. Honestly, if I were doing notes right now by myself at home, I would probably pause here and come back to this in like 30 minutes because I would need a little break. But viruses are not living, right? And they tell you here to review the characteristics of living things. So they do have DNA, which is great, but they cannot reproduce on their own, right? They can't do mitosis, they can't do meiosis, they don't have a cell wall, they don't have a cell membrane, they're literally a protein that holds DNA or RNA. Sometimes they're RNA viruses. In fact, those are the hardest ones to prevent. They have all these different shapes, right? This is my favorite one because it looks like an alien invasion. In fact, let's look at one. They really do look like spaceships invading the cell. That's a weird picture. Let's see if we can find a good one. Yeah, this is a great picture of what viruses look like. Let's see if I can find one on a cell. Dang, there's no good pictures of these really. This one's okay. But basically what they do is, it looks like a video game. They sort of land on a cell. They inject their DNA or RNA into the cell, and then they use the cell to make thousands of copies of themselves, right? So they turn your cells into factories to make millions and millions of copies. And then once they're done making copies, they explode your cell, and then they invade other cells. And during that exploding process, when cells are exploding, that's when you're going to feel symptoms of a virus, when you're not going to feel good, right? And some viruses can last for months, so that's fun. Okay. I'm not going to go too much into viruses. In fact, we'll talk about them in the future, but that's my little spiel about them. They are not living, so they shouldn't be in this unit, excuse me, this lesson about living organisms. But they are. All right, so make sure you take notes about viruses. Otherwise, right, make sure you have notes written down. Hopefully you have like a notebook, because I just did this on paper. But hopefully you have a notebook where you actually are keeping these notes so that you can go back and review them. Because when you take a test, right, or a quiz, or you do an assignment, you'll have your notes to use. Okay, that was a lot of talking. So now we'll open it up for questions. Oh, also, I just wanted to say one thing too. There's no correct way to take notes. Like, look at my math notes. This is how my math notes look, which most people would probably not be able to understand how do I make this focus? What this is that they're looking at. But that my notes are just literally me writing out a million things. I write all over in the margins at the top. I draw pictures. I write weird stuff. So remember, your notes are your notes. They might not look how mine look, which is totally fine. You might have more written down. But really what I want you to get at is don't write everything in the lesson. It's a waste of your time, and probably at least a third of the information there is something that you don't need to write down. All right, so questions from people, if we have any. You've already done the assignment. Do you have to be here? Yes, because I added extra stuff. So the exit ticket for this is going to be 3 questions. First one is going to be, why can it be important for scientists to organize living things? So really, what's the point of creating a system like this, or creating a cladogram, or creating any of these things? Why would we want to do it? The answer might be we don't need to do it. You could argue that. Then what I'm going to have you do is choose 2 of the following phyla, right? If you remember from our kingdom, phyla, class, order, family, genus, species. These are some phyla, and I want you to pick 2 of them and describe them in detail. What makes them a phyla? Why are organisms in that phyla? And then the last one is a cladogram one that's really hard. These are the 4 animals / living things that I picked. I want you to figure out how to make them into a cladogram. So picking traits that will separate them out. Okay, so I posted the exit ticket on Canvas already. So you can see it there, and I'm going to be posting this video. Oh, come on, thing. Move. There we go. So if you go into your modules and you go to virtual session on 3/13, oh yeah, this is a friendly reminder that the dates are not going to line up for those virtual session exit tickets. I tried really hard to make them line up, but almost everyone's on a different date at this point, so it's not going to line up. So I posted the exit ticket there, right? So you can submit it and you can view it there. Otherwise, I'm going to post the PowerPoint, and I'm going to post a video of this. And I'm also going to take pictures of the notes and post those as well. And otherwise, if you don't have any questions, we're done. So we'll stop sharing here. Thank you for those of us who took time out of our day to come to this. I'm going to take a pic of who's here. All right. And I will talk to you guys soon. And thank you for coming.